welcome back to the channel. In today's video, we are going to simplify glycolysis, which is the first and most fundamental pathway of glucose metabolism. By the end of this video, you will understand what glycolysis is, why it is important, and we'll go through each reaction step by step in very clear and easy way. The term glycolysis comes from Greek word where glycose means sugar and lysis means breakdown. So the glycolysis literally means the breakdown of sugars. It is a universal metabolic pathway, meaning it occurs in almost all living cells, regardless of whether oxygen is present or not. The complete pathway of glycolysis was worked out in the year 1940, since it was described by Embden, Meyerhof, and Parnas. It is also known as Embden-Meyerhof-Parnas pathway, or simply EMP pathway. Now for glycolysis definition, glycolysis is defined as a sequence of enzyme catalyzed reaction in which glucose is converted into pyruvate under aerobic conditions or into a lactate under anaerobic conditions with the generation of ATP. So it means glycolysis occurs in cytosol of the cell and does not require oxygen, which makes it extremely important for tissues like red blood cells that lacks mitochondria. It's also essential for brain, which depends mainly on glucose for energy. In addition, the intermediates of glycolysis are used for the synthesis of amino acids and fats, and the reversal of glycolysis leads to gluconeogenesis. Now that we understand what glycolysis is and why it is so important, let's begin with the actual reactions of glycolysis. Before we start explaining each reaction, it is very important to understand the overall structure of glycolysis. Glycolysis is not a single reaction. It is a sequence of 10 steps, and these steps always occurs in same order. To make it easier to understand, the glycolytic pathway is divided into three phases. The first phase is energy investment phase, also called as primary phase. In this phase, ATP is used to activate glucose. In second phase, it is called splitting phase, where the 6 carbon of glucose molecule is split into 2 or 3 carbons molecules. The third phase is energy generating phase, also known as the payoff phase. In this phase, ATP and NADH are produced. So remember, glycolysis have 10 steps arranged in logical sequence, and understanding these phases will make each reaction much easier to follow. Also, I have color coded the sections where the ATP is used, it will be red, and where the ATP is generated, it will be blue. Let's now look at the first reaction of glycolysis in very simple way. On the screen you can see the structure of glucose, which is 6 carbon sugar. Now focus on the 6th carbon of glucose. This carbon has an hydroxyl group attached to it. In this step, the enzyme hexokinase helps to transfer the phosphate group from ATP to the 6th carbon of glucose. As the ATP donates the phosphate group, it gets converted into ADP because ATP is involved here. I have marked the step in red. After the phosphate group is added, glucose is now converted into glucose 6-phosphate. This phosphorylation step is important because it traps glucose inside the cell and commits it to the further metabolism. It is an irreversible reaction. So remember, when you see the red in this pathway, ATP is involved. Now let's move on to the second reaction of glycolysis, which is isomerization reaction. In the first step, glucose was converted into glucose 6-phosphate. This molecule is impermeable to the cell membrane, so it must continue through the glycolytic pathway. In this step, glucose 6-phosphate is converted into fructose 6-phosphate by the enzyme phosphohexoisomerase. Here, there is no addition or removal of atom, only the structural rearrangement takes place. Glucose 6-phosphate is aldose sugar, whereas fructose 6-phosphate is ketose sugar. This conversion is important because fructose 6-phosphate can be easily phosphorylated in next step of glycolysis. This reaction is reversible and it does not involve any ATP. So there is no red marking here. Now we can come to the third step of glycolysis, which is phosphorylation reaction, and this is the most important regulatory step of this pathway. In this step, fructose 6-phosphate is phosphorylated to form fructose 1,6 bisphosphate. This reaction is catalyzed by enzyme phosphofructokinase 1, which is also known as PFK1. Here, a phosphate group from ATP is added to the first carbon of fructose 6-phosphate. As ATP donates the phosphate group, it gets converted into ADP. This step is marked in red because ATP is used here, making it a part of energy investing phase. This reaction is irreversible and serve as the rate limiting and regulatory step of glycolysis, which means the overall rate of glycolysis is mainly controlled at this step. Now that fructose 1, bisphosphate has been formed, the glucose molecule is fully activated and ready to be split into 2,3 carbon units. Now we enter the splitting phase of glycolysis, which begins with reaction 4. In this step, the 6 carbon compound fructose 1, 6 bisphosphate is split into 2,3 carbon molecules. This reaction is catalyzed by enzyme aldolase. The two products that are formed are dihydroxyacetone phosphate, that is also called as DHAP, and glyceraldehyde 3 phosphate, which is G3P. So from 1,6 carbon sugar, we get 2,3 carbon compounds. This reaction is reversible and does not involve any ATP, which is why there is no red marking in this step. Now let's move to the reaction 5 of glycolysis, which is again an isomerization reaction. In the previous step, fructose 1,6 bisphosphate was split into two 3 carbon compounds, dihydroxyacetone phosphate or DHAP and glyceraldehyde 3 phosphate or G3P. However, only glyceraldehyde 3 phosphate can continue further in glycolysis. So in this step, the enzyme triose phosphate isomerase catalyzes the reversible interconversion of DHAP into G3P. As a result, from one molecule of glucose, we finally obtain two molecules of G3P, which will now enter the energy generating phase of glycolysis. From this point onwards, glycolysis enters the energy generating phase, which is also called as payoff phase, where ATP and NADH are produced. Now we enter the reaction 6 of glycolysis, which marks the beginning of energy generating phase. In this step, glyceraldehyde 3 phosphate or G3P is converted into 1,3 bisphosphoglycerate by the enzyme glyceraldehyde 3 phosphate dehydrogenase. This reaction is very important because it involves an oxidation of G3P. During this process, NAD plus is reduced to NADH plus hydrogen ion. At the same time, a high energy compound 1,3 bisphosphoglycerate is formed. I have marked this step in blue because there is the first energy generating step of glycolysis where reducing power in the form of NADH is produced. Also remember, we have now two molecules of G3P plus glucose. This reaction occurs twice for each glucose molecule. Now we come to the reaction 7 of glycolysis, which is very important step because this is where ATP is actually synthesized. In this reaction, the high energy compound 1,3 bisphosphoglycerate is converted into 3-phosphoglycerate by the enzyme phosphoglycerate kinase. During this process, a phosphate group is directly transferred from the substrate to ADP, resulting in the formation of ATP. This type of ATP synthesis is called substrate level phosphorylation because ATP is formed directly from the substrate without the involvement of electron transport chain. So I have marked this step as in color blue to indicate the ATP formation. This reaction is reversible and it is a classic example of kinase reaction where a phosphate group is transferred between the molecules. Also remember, since two molecules of 1,3 bisphosphoglycerate are present per glucose, 2 ATP are formed in this step. Okay, so now let's move to the reaction 8 of glycolysis, which is isomerization reaction. In this step, 3-phosphoglycerate is converted into 2-phosphoglycerate by the enzyme phosphoglycerate mutase. Here, there is no addition or removal of atom. The reaction involves only rearrangement of phosphate group from third carbon to the second carbon of the molecule. This reaction is reversible and does not involve ATP, but it is important because it prepares the molecule for the formation of another high energy compound in next step. So the next step is reaction 9 of glycolysis, which is dehydration reaction. In this step, 2-phosphoglycerate is converted into phosphophenol pyruvate, also called as PEP, by the enzyme analase. During this reaction, a molecule of water is removed, which result in the formation of phosphophenol pyruvate, a high energy compound. The enzyme analase requires magnesium ion or manganese ion as cofactor for its activity. This reaction is reversible, but it is very important because the high energy compound formed here will be used to the produce ATP in the final step of glycolysis. Now we have come to the final step of glycolysis, which is again substrate level phosphorylation reaction. In this step, the high energy phosphate group from phosphophenol pyruvate or PEP is transferred directly to the ADP, resulting in the formation of ATP. This reaction is catalyzed by the enzyme pyruvate kinase. Pyruvate kinase requires potassium ion along with either magnesium or manganese for its activity. Since ATP is formed directly from the substrate without the involvement of electron transport chain, this step is also an example of a substrate level phosphorylation. This is an irreversible reaction and the final product formed is pyruvic acid, also known as pyruvate. With this step, the glycolytic pathway is complete. So now let's calculate the net gain of glycolysis in simple way. First, ATP is used in two steps, step 1 and step 3. So 2 ATP are consumed. ATP is formed directly in two steps, step 7 and step 10, producing total of 4 ATPs. Next step is 2 NADH molecule are formed in step 6. Using this modern value, 1 NADH produce 2.5 ATP. So 2 NADH produce 5 ATPs. So overall, 4 ATPs are formed, 2 are used, and 5 come from NADH, giving a net gain of 7 ATPs per molecule of glucose. So that's glycolysis done step by step without overcomplicating it. We have completed the glycolysis from start to finish. If this video helped you understand the pathway better, do like the video and subscribe for more simplified metabolism videos like this. Thank you for watching and I'll see you in the next one.